leave early. Yeah.
If we could leave early. So if we want to leave early, we have to get started now. Okay. So today we start on the last section in this chapter. We're going to talk about probability and statistics. It's not going to be tested on. The first few slides are still material we've already covered. It's just review and a little bit of follow-up. That will be on the exam. There will be a point at which we switch today to probability and statistics. That won't be on the exam. And I'll tell you when that transition happens. So first, go ahead.
Would any of the first test material be on the second test?
Will first test material be on the second test? Absolutely, because we're building on what we already know. So I'm not specifically. There won't be questions specifically about that content, but it's incorporated into questions that are on this test. So because I've been using this hypothetical example about green fluorescent rolling worms for so long, I wanted to make sure that we actually explicitly answered the question, what's the best way to cross different strains of worms together to produce worms that do the thing that we want to do? So last class we started this conversation. And we had one student at least indicate that this might be a cross to perform. You have the roller worm, one of the strains that I provide you to do this cross. It produces only gametes that have a minus at the first gene. That's the roller gene. Right, that's the order we're talking about the genes. Left to right, roller first, GFP second. So a worm that's homozygous for a mutation in the roller gene only produces gametes that carry that. And those gametes from that individual have to carry the plus, the wild type allele, not fluorescent, at the GFP gene. So there's only one type of gamete that individual can produce, so that there's only one column in the Punnett square. Don't need all four, because they would all have the exact same thing written 4 different times. And if you look at the gametes that the other worm that's provided, the GFP strain, how many types of gametes does it produce? It has to have a plus, because this individual only has pluses, so all of the gametes have to have pluses for the roller gene, their wild type versions, the wild type alleles. So why are there 2 rows? What is it about that GFP worm in the upper right that makes us need to have 2 rows in the Punnett square?
It's heterozygosity of fluorescence?
Yeah, it's heterozygous here. It's got a plus and an asterisk. So it's got 2 copies of this chromosome. Half of the gametes are going to get the plus, and half are going to get the asterisk. So that's why there are 2 rows here. Which of Mendel's laws is that? Half of the gametes get one version of the chromosome and the other half get the other version. First law, which is the law of segregation. The segregation of alleles into separate gametes. Very good. So if we actually write out the genotypes, start with the first gene, the roller locus. It gets a plus from the parent on the left and a minus from the parent whose gamete is on top. Semicolon, and then asterisk and plus at the GFP gene. So what's the phenotype of this worm? What does it look like if you looked at it under a microscope? Does it roll or does it not roll?
It doesn't roll, it's fluorescent.
Who thinks this is a roller worm? Raise your hand. Who thinks this is not a roller worm? Yep. If even one person raised their hand, that would have been a yes. Nobody raised their hand for the first. So it only has one copy of the mutation, the minus, the roller gene, so it's not a rolling worm. But is it fluorescent? I want to see a lot of heads nodding. Yes, thank you. Okay. Because it's got one copy of the gene that makes green fluorescence, the asterisk. So this is a non-rolling green fluorescent worm. What about the one below it? Plus over minus, plus over plus. So it's also not a roller. It doesn't have 2 minuses at the first gene. And it's also not green fluorescent, so this looks like a wild type normal worm. So if we have these 4 strains, if we've got the roller worm, if we've got the GFP worms, and now we have 2 different types, 2 different genotypes of F1s, we've got 4 different genotypes of worms here. How would you set up a cross to make the worms we're trying to get? Green worms that are also rollers. So is it better to mate siblings? Let's focus on these 2 questions. Is it better to mate siblings together, mate F1 individuals together, or would you mate kids with their parents? Only because we're trying to create green roller worms, not because we're trying to do weird things.
Very unsocial.
But yes, yeah, I'm sorry. I should be more sensitive about these sorts of things.
But yeah, I would go with the parent and child. I would go with 1 and 3.
Okay, so we've got one proposal to cross a worm with genotype 1 and a worm with genotype 3. Why would that make sense? If we're trying to make worms that are green fluorescent and roller, which one's harder to do, does it seem? To make a roller, why? Because you have to have the correct allele at both alleles, sorry. You have to have 2 alleles, the recessive alleles, to get a roller. So if it's recessive, it's harder to find individuals that have that unique combination, a minus from mom and a minus from dad. Why is it relatively easy to make a green worm? Same answer. Yeah, you only need 1 asterisk, 1 star, to make a green worm. So let's worry more about how we're going to make crosses between individuals where we're assured that we're going to get minus over minus. Of all of these combinations of crosses, what does that look like? Which is the worm we have to use if we want to make sure that we're optimizing our chances of having minus alleles sent into the next generation? Yeah, I see a few people raising 1 finger. I hope that's the index finger. Right, we want to use worm 1 because it is guaranteed to contribute a minus allele to the next generation. We only have a 50% chance if we use worm 3 or worm 4. Which one do we absolutely not want to use? Worm 2, which has no minus alleles at all. So it's got no possibility. If you set up any cross with worm 2, there's no chance that its kids will be rollers, because they're guaranteed to get at least 1 wild type allele from that parent number 2. So that looks like, I drew a couple of examples here to get us going. What we just talked about looked like this one I'm circling in red. Cross between an F1 individual and one of the parents, the roller. So let's see if we're right. How do we set up this Punnett square? If this is what's underlined in blue here, if that's the cross we're making, so you want to put the F1 genotypes across the top?
Yeah.
Okay, so we've got half of the gametes get plus, half of the gametes get minus, and then that's the first gene. Then what do we put also in these gametes? Now we have to talk about the GFP gene. So yeah, we could put a star and then a plus, and a star and a plus. So we made the 4 possible combinations of gametes that are produced by a dihybrid individual.
Would it make sense to say for the top, instead of doing plus semicolon star underneath it, could you do plus dash star, or would that be indicative of it?
So we have to be very careful about terminology because, so you could use a hyphen, but geneticists use a semicolon.
I should have said dash.
No slashes. So slash represents two different versions. So we've got some slashes up here. This is a great question, by the way. Same gene. So slashes separate the two alleles of the same gene. You have to be very careful about this. And the semicolon separates what we were talking about, separate genes. A gene that makes rolling worms, a gene that makes fluorescent worms. The semicolon in between them. Okay. So what goes on the y-axis in this Punnett square? We've got that parent, its alleles, its gametes go on the y-axis. What do I write there? Down the side. If it's minus over minus at the roller gene, what allele does it put into all of its gametes? A minus. Okay. What about the next gene that we need to talk about, the GFP gene? It's homozygous. It's got two copies of the wild type or plus allele, so we fill in pluses. So it's minus plus, minus plus, minus plus, minus plus. Again, we've reached that point where if you start with a pure breeding parent, it's going to have the same combination of alleles in all of its gametes. So we don't even really have to write in those bottom three rows. Saves us a lot of time. We don't have to fill in any of that. It gives us exactly the same information as that first row will. So if we fill this in now, we can find out what genotypes we have and whether or not we actually succeeded in making any green fluorescent rollers. Did we? We've got 25%, we've got 4 different genotypes in there. Quarter of the offspring will be each of those 4 squares. Which one is the green fluorescent roller? First one, second one, third one, or fourth one, left to right? Third one. We've got a genotype that's got 2 minuses at the roller gene and an asterisk over plus. Right, that was one of the 2 different genotypes we're looking for up here in the box in the upper right-hand corner that can make green fluorescent worms. Yep.
So when you're doing a dihybrid cross,
Yep.
so you know we're doing a monohybrid cross, we're looking at one gene.
Yep.
One trait.
Right.
From one parent, we put the 2 traits, the 2 alleles on the top and the other, from the parent, the other 2 alleles on the left.
So if you
It seems like you're combining those 2 that are on the top and on the side of a monohybrid cross on the top. Is that one, that they get those
so in this case,
combination?
the pluses, let me switch to a different color. So these pluses, how did I get what I wrote right there? Is essentially rephrasing your question. Where did that information come from? So those both came from this F1 parent. So we're putting the F1 individual's gamete types across the x-axis. The symbol that's on top, the plus, came from the first gene we're talking about. Right? We go from a diploid F1 individual to a haploid gamete. So you get to pick a plus or a minus. You put that in the gamete. The second thing that's written there came from the other gene we're talking about. You either get a plus or a star. So if you've got an F1 individual that's got a plus and a minus and a plus and a star, there are 4 different combinations of those that you can make, and that's what's shown across the top. By the way, which of Mendel's laws is this? This is the second law, which is independent assortment, which means if you've got 2 genes, then whichever allele of one gene you put into a gamete has nothing to do with whatever you put in the second gamete. That's why, despite the fact that this gamete has minuses, half of them have an asterisk and half of them get a plus at the second gene. So we've got one GFP positive and rolling worm right there. Happy? We set out what we wanted to do, but how much money are you going to make at the rave when you go sell this worm? Not very much, because you got one worm, or in this case, a quarter of your worms. You set up a cross, you made a quarter, 25%. This is not very good return on your investment. You throw out 3/4 of the worms you make because they don't have the traits you want. What would be better? The real goal is to make a pure breeding worm that's green fluorescent and a roller. Which genotype are we looking for? Right, we're looking for the other genotype that makes a green fluorescent roller that we haven't seen yet in this cross. Minus, minus, asterisk, asterisk, a pure breeding homozygous at both locus, both loci genotype. Will we get one of those if we do the cross on the left? The Punnett square we haven't filled in yet. If both parents have a minus allele and both parents have an asterisk allele, is there any chance that we get all 4 of those together in one worm? So I'm going to set up the Punnett square. You tell me where the worm is that we're looking for. So we,
Third row, third column. In the third row and the third column, that's where you're gonna find.
so there's the combination of genotypes we're looking for. Minuses with asterisks. So guarantee that the one individual out of 16 that gets that combination of gametes from both of the parents is going to be the homozygous true breeding GFP roller, green fluorescent roller. Will any of the rest of the squares have 2 minuses and 2 asterisks? That's the only place in that square of 16. Didn't even have to fill in all 16 squares to figure this out. Nor will I ask you ever to fill out a 16 square Punnett square during an exam. There's always shortcuts like this that you can take, so there will never be a question where you're actually explicitly told, please fill in all of the allele symbols for all 16 squares. So which is the best way to do this? Depends on the goal.
I mean, you have to do another breeding too.
So then you have to take that individual and self it, or mate it with a sibling that's got the same genotype. And then you make 100% green fluorescent rolling worms, and then you're in business.
And you can do it over.
Then, yep, then you take one of those offspring and you keep going. Thank you very much, all of you, for those of you that contributed to the PCR genotyping analysis. I was really impressed with a lot of the submissions that came in, and so I want to go through and highlight a few of them, because this will be on the exam. So this is content that could be tested on. We've already talked about it on one exam, but now we're looking at real PCR gels, analyzing the data. So I liked this example, A, because this student tabulated how many of each of the different genotypes they interpreted that they saw. So in the top half of the gel, 7 homozygous AF16, 2 heterozygotes, and so forth. That was one of the questions I asked you to do. Now, what's at the bottom here? The Punnett square. Why does this student write out the Punnett square?
Percentage.
So we want to know the percentage of, what does the Punnett square tell us? Why do we ever use a Punnett square? Right. If we set up a cross, how many of each genotype or phenotype do we expect to see? So this is recreating the Punnett square to figure out, we observe that many AF16 over AF16, that many AF over HK, and so forth, do our expected values match our observed values?
No.
They don't look like they do. What about this one, though? This is a little bit different. So this student came up with slightly different numbers of counts, and you'll see there's a question mark here. 3. Some of these genotypes were maybe hard to read. The bands are really faint. You don't know, is it one large band, one small band? Is it heterozygote or is it not? The thing that I really liked about this is the third submission rather, the one on the right half of the screen. What's new here that we haven't seen before? So we've got a list of all the genotypes for each of the rows. We've got some question marks. What do we do with the genotype that's got 3 different bands? We expect to see a 1 to 2 to 1 ratio. What else is in the figure on the right that's essential for figuring out if the observed and the expected values match? Haven't seen this yet today. What's in there with the wavy lines? Is that the symbol for Aquarius?
It's 3.
Oh, is it 3 wavy lines for Aquarius? I'm an Aquarius, I should know. Go February. What are those? Approximately equal to. What do those numbers represent? Those aren't frequencies. This is not a Punnett square. What are those numbers?
The number of individuals.
They're expected numbers of observations. That is not frequencies. What we looked at in the Punnett square was 25%, 25%, 25%, 25%, a 1 to 2 to 1 ratio. So up to this point, everybody that submitted, that I'm showing you, we're talking about when we expect something, it was in frequencies or percent. Here, this has been converted into actual numbers of, in this case, wells on an agarose gel. How many wells did we expect to observe AF16 over AF16? About 5. About 10 for heterozygotes, about 5 for the HK over HK genotype. Having the number of expected observations, not a frequency or percentage, is absolutely critical. This is the number 1 mistake that gets made on chi-square tests. If you have an observed number of AF16 homozygous counts and an observed number of heterozygous reads, you have to compare those to your expected numbers, not expected frequencies or percentages. So this student actually explicitly shows us the formula for how to do that, how to convert our expectation based on a Punnett square into the observed number of, the expected number of observations. So how do you do it? You multiply the percent, so those are the percentages that I just circled in red there in the middle. Those are the percentages that came from the Punnett square. 25% AF over AF, 25% HK over HK, 50% heterozygote at the bottom. You multiply that by the total number of observations. In this case, the total number of wells that we read genotypes for, which in this case was 22. So there are 22 wells on this gel that we can interpret genotypes for. So a quarter of those 22 should have been AF16 over AF16. That would be 5.5. And a quarter should have been homozygous for HK104, and a half should have been heterozygous. Is anybody uncomfortable with this? You should be. So please tell me why. How do you read 0.5 wells? What does a 0.5 mean when you have an expected number of observations? So what do you do with this? Do you round it or do you not round it? Very important. Important thing 1, work with expected numbers, not frequencies or percentages. Important thing 2, keep it as it is. Because the total of that column has to equal the total number of observations. We looked at 22 wells on a gel, so the expected number has to total exactly the same. And the thing that I really liked about this, the last one, interpretation. So this student went above and beyond by saying not only did I look at expected and observed, but I compared the two, and it didn't look like they matched. And that's what the chi-square test is for. So there's no chi-square test here yet. That's what we're starting today. But the point is, what does this say? This does not look like a typical F2 generation when two heterozygous organisms are crossed. Look at this. Actually proposed, I suspect that these data actually came from this type of a cross. Blows me away. So thank you to the student that turned this in. This was really inspiring to see. So here I've summarized those example slides I just showed you and their observed counts. Mine's on the left. I counted 17, so this is me. I counted 17, 2, and 3. Was there an agreement? Two of us agreed on 17, 2, and 3. Two others of us agreed on 16, 3, and 3. So an important point that I'm always making through the term, there's not always one correct answer. It depends on how you read and interpreted that gel. This is true in science in general. When you have a picture and you're trying to interpret visual evidence, there's always going to be some variation. So there isn't a right answer here. We could go with a consensus answer if we wanted to. So when you compare any of those 5 columns that have observations and the expected column on the far right, 5.5, 5.5, and 11, how many of you would agree that any of our observations match, are the same as our expectation? Be brave, raise your hand. Do they match? If so everyone's going to raise their hand when I ask the other question? Who thinks they don't match? Nobody's awake, nobody gets attendance points for the day. Who thinks they don't match? Okay, well, we've got a few people earning attendance points. They don't match. But we're scientists. So we have to agree on a metric, something quantitative, something we can measure, that helps us decide how much do they match. There has to be a threshold where we say these values are similar enough, the observed values are similar enough to expected that we're going to agree that they match. There's going to be a line there where we say if the values look this much different, we're all going to agree that they're not the same. That's what the chi-square test is for. Right. So that's the main goal for today. Okay. First question. You've already seen that expected and observed values don't match. They didn't definitely in this PCR gel. But even in Mendel's data, you calculated these data. One of you sent me this spreadsheet. All of yours should have looked essentially the same. We calculated the F2 ratio over there. We expected 3-to-1 ratio. For the top cross, we observed 2.95 to 1. For the bottom cross, it's a little more than 3 to 1. So even Mendel didn't get the exactly expected values. Why not? Why didn't Mendel see exactly 3.0000000 3-to-1 ratio? Something happened. Mutation. Could be mutation. Probability. So what's the likelihood that he looks at, holy crap, what is that, 8,000 plants or something like that, approximately? What's the probability that he looks at 8,000 plants and sees exactly a third or a quarter of them have one phenotype and 3/4 have the other? it's pretty good, and his answer is pretty close to 3 to 1. It's just slightly off, but that's random chance. By chance, certain individuals with certain genotypes maybe didn't survive for him to look at, and so it's not quite 3 to 1.
Dr. Rob?
Yeah.
Give a classic example, they say like, when you flip a coin, we all know it's 50%.
Oh, I didn't bring my coins. Oh well. Yes.
But if like, if you flip the coin 2 times, you can get
Heads.
heads 2 times.
Right.
That doesn't mean you don't get 50. The more times you do it, you should end up getting that ratio.
So given the fact that if you flip a coin twice, you should see 1 heads and 1 tails, and we're going to talk about this in terms of sex determination in just a minute, so stay tuned. Right? How would you improve your probability of actually observing what you expect if you're flipping coins? What do we as geneticists have to do? What did Mendel do to try to improve his observations? How many times are you going to flip the coin? Twice? 100 times? What do? What's practical? You flip it as many times as you have time for before it's lunch, or something like that. The more observations we have, the closer we should get to what the expected frequency is. And that's why Mendel's data are pretty close to 3 to 1, because he looked at something like 8,000 plants. Okay, so here's where it gets personal. How many of you think that I am male? Raise your hand. I won't get offended. How many of you think I'm female? Okay, nobody's brave enough to raise their hand. Okay, so here's our null hypothesis. That is a fancy way of saying an expectation. Dr. Ross is male. That's going to be our hypothesis. We're going to test this hypothesis. Here's the observation. Well, my wife isn't part of the observation. This is my family, my wife and my 2 daughters. So it turns out in humans, you probably already know, that males have an X chromosome and a Y chromosome. And that's how we become males. If you get a Y chromosome, you're male. If you don't have a Y chromosome, you're not. So if you mate a human male and a human female together, half of the male's gametes get the X chromosome, the other half get the Y, and the female has 2 X chromosomes. So what's our expectation? If anybody has a kid, what's the probability of having a male? 50-50. Half of these squares, 50% of the volume, the area of the table is XY. So half, 50% male. And this is a good thing too, because if we had anything other than a 50-50 sex ratio, if there were more girls than boys or vice versa, then our species would eventually go extinct because there would be eventually none of one sex, which would be bad, presumably. Well, for us anyway. All the other species on Earth would probably appreciate it. So that's expectation. 50% of kids should be male, 50% female. What's the observation? 2 females, 0 males that I've produced in my lifetime. With the help of my wife, thank you very much. In no small part, I should say. So, do I have any evidence of having a Y chromosome? That is, am I male? I have not yet produced any biological evidence that I have a Y chromosome because I have no sons. It's true. There's some other evidence, facial hair and deep voice and all that sort of thing. I could be faking it though. So, chi-square test. We have observation and we have expectation. If I have 2 kids, we can't use frequencies or percentages, right? We have to use number of observations. So if I have a total of 2 kids, how many males and how many females should there be? I should have had, if I'm following Mendel's laws, 1 male and 1 female having 2 kids, 50% chance. What have we observed? We've observed 0 males and 2 females. So to do a chi-square test, well, I don't need to write out the formula. Presumably you watched the video that I posted that you were supposed to have watched for today. It didn't look like there were 75 views. You're okay because this is not on the test, which is I'm assuming why most of you didn't watch it. But do watch it. You'll see what the chi-square formula is. If you don't know, it's observed value minus expected value. So 0 minus 1 squared over the expected value. And you do that for every category. So here there are two categories, males and females. There are two types of phenotypes we're counting. So that's for sons, observed minus expected squared over expected. And then we do daughters. Observed, 2, Natalie and Audrey. 1 expected, don't know what her name would have been, over expected. And that gives us the statistic value we call the chi-squared value. And in this case, 0 minus 1 squared over 1 is 1. 2 minus 1, forgot the squared, thank you, is also 1. So this is 1 plus 1, which equals 2. So the chi-square value is 2. We also need to know one other thing to ask whether or not I have evidence of a Y chromosome or not. Degrees of freedom. DF here in the table. And that's, as it says in the video, the number of categories minus 1. So we're looking at 2 categories, 2 sexes, male and female, subtract 1. So the degrees of freedom is equal to 1. So those are the 2 pieces of information we need to figure out if I've got a Y chromosome. Chi-square value of 2, 1 degree of freedom. So how do we use this table to actually figure out what the probability, the p-value, is? And that's what these subscripts are, the numbers. Those are the probabilities. So that's our goal, to figure out the p, or probability value, that the hypothesis is true. So where does a value of 2, we have 1 degree of freedom, so we read from left to right, where do we hit the number 2? It's somewhere in between there and there, somewhere between 0.016 and 2.706. So the chi-square value we calculated is in that range, which corresponds to p-values, probability values, that are between 0.9 and 0.1. So that's calculating the chi-square value, calculating the degrees of freedom, and then looking up the associated p-value in the header of this table. What does it mean that the p-value is between 0.9 and 0.1? This is the really important point. It's not chi-squared, no, it's not t. It's not chi either. This isn't tai chi, this is chi-squared. What does it mean that the p-value is between 0.9 and 0.1? It means there's a between 90% and 10% chance of something, 0.9, 0.1. 90% to 10% chance that the null hypothesis is true. So that's somewhere between a 90 and 10% chance that the null is true. What was the null hypothesis? That I'm a male. So thank goodness, you can go home and tell your friends that there's a 90 to 10% chance that your professor of genetics is actually male, despite that that's what you thought anyway. So that's how you use a chi-square test, and this is how scientists have decided that we're going to determine whether or not the observed values and the expected values are different or not. The magical threshold is p-value of 0.05. If your p-value is smaller than that, to the right anywhere, that means the observed values are not equal to the expected values. There's a statistically significant difference between your observation and your expectation. Is anybody here from a family where the observation is absolutely the same as the expectation? So how many sons and daughters? Two sons, 2 daughters? Okay. Where does chi-squared value of 0 show up on the table? Now I've written on top of it.
P-value 0.995.
It's all the way here on the left. Right? Where the chi-square value is 0, the p-value is 1. 100% chance that the null hypothesis is true. So you've proven to yourself that, hey, when the observed and the expected values match, you get a chi-squared value that is really small. Really small chi-square value means your probability of expected and observed values matching is very high. So that's what I'm after. Maybe if we have a third kid, a son. So what happens to the chi-square test if your observed values, so here's males, females, we observed family of 4, well, family of 4 kids anyway. 2 sons and 2 daughters. We expect to see, in a family of 4, 2 sons and 2 daughters. What happens when you do the chi-square test here? Observed minus expected squared over expected plus observed minus expected. Wow, that's a lot of 2's. If you subtract 2 from itself, it's 0. If you square it, it's 0. If you divide it by something, that's still 0. So what's the chi-square test statistic? 0. When the observed and the expected values match, this is how you can do this on the fly if you ever forget how to interpret a p-value. What does it mean if p, is it p greater than 0.05 or less than 0.05? That's the one I'm looking for. When you're observed and your expected values, you know, you're certain they match because they do. You get a chi-squared value of 0. Now we could do this also for the CBM 56 data. We will at some point. So we have observations that all of you helped generate by looking at our PCR gel and telling us which genotypes there are. And we'll follow up with that at a later date. But before we go, I've got one other thing to say. So we'll carry on with that at some point. I am trying to make good on my promise that I would let you out early if we started. So the only thing you really need to do for Monday is to come with questions about the test. Monday's review session, Wednesday's the exam. And I've already posted the study guide and the video on YouTube that is the key. And I just wanted to point out, in case anybody hasn't noticed on YouTube, that I've been putting tables of contents in the information section for each video. When you go to the video, you poke the thing that says show more. It gives you at which point in the video each question begins, or the answer to each question begins, so you don't have to watch a 50-minute video to figure out where the answer to the question you need an answer to is. Just click on the time that's associated with the question that you're interested in, and it takes you right there. You get that question answered. So, for studying for the exam, I suggest take the practice guide like a mock exam. Don't look at the key yet. Take it as a mock exam, see where you stall on answering any one of the questions, then go look at the study guide, see where you went wrong, bring questions, do that over the weekend, bring questions about what you don't understand at that point to class on Monday. One question, please. The study guide is blank. Maybe that tells you something about the exam. No, it does not tell you anything about the exam. I will try to fix the problem if I can. Thank you. Okay, see you next time.